Dear learners, welcome to the learning session. Today we'll discuss low temperature in context of preservation of foods. As by now you are aware that food is a perishable commodity and it is a basic need of life. So we store food. There are different methods of storage of food. Low temperature, which is used and very commonly used for storage. So low temperature has a connection in terms of food preservation, and there are certain techniques which are also applied where we process the food using low temperature. Low temperature applications in food, they are known as chilling and freezing prominently. And in relation to food, the low temperature is addressing the challenge or spoilage cause, that is microorganism. If you recall the basic microbiology knowledge, the microorganisms are classified as mesophiles, psychrophiles, psychotrophs on the basis of what? On the basis of the temperature which they need for their growth. And microbes are challenged to the food. If they grow in the food, there is likelihood of the spoilage. Mesophiles, they need a temperature in a range of 20 to 45 degrees centigrade, where optimally they can grow 30 to 39 degrees centigrade. There could be lot many spoilage organisms could be there in the mesophiles. Psychrophiles, and today we are talking in terms of low temperature, so psychrophiles and psychotrophs both are very important, which are psychrophiles, particularly also known as cryophiles. They have optimum temperature which is almost around 15 degrees centigrade or even lower. And maximum side, if you know, they can tolerate up to 20, and lower side they can go up to 0 degrees centigrade. However, psychotrophs, they are more facultative. Means they have ability to grow at as low as 0, but usually optimal maximum growth temperature that goes a little higher, which is 15 or above up to 20. Another important thing is that metabolic activities, which is of microbes, or you can say any biochemical system, if it is there, exposed to the low temperature, that activity, biochemical activity or metabolic activity is slowed down if the temperature is low, and generally almost stopped even if it is coming closer to the freezing temperature. And at every 10 degrees centigrade fall in temperature, up to certain extent, this slowdown would be 2 to 3 fold in terms of its rates of reaction. Psychotrophs and psychrophiles, either bacteria or mold, they are able to grow at low temperature, as we have seen, I've just explained. It is important to look in terms of low temperature storage or low temperature preservation of the food. The temperature which is called 7 degrees centigrade acts as a boundary below which the mesophiles usually, or most of the mesophiles, they stops grow. So it means one challenge is taken care, but still psychrophiles, psychotrophs, they may pose a challenge to the food items. And for that, we need to study in detail how to go for low temperature applications in terms of the food preservation. Molecular mobility, which is actually depressed, and there how the chemical reactions or the biological changes which are taking place in foods, they are slowed down. Or equally, this is applicable to the microbes. So, what is the principle here? The principle here is microbial growth is taken care, biochemical changes are taken care, chemical changes are being taken care. In a way, they are slowed down. And important to look in terms of what? Some microorganisms like psychotrophs, there are certain bacteria, most commonly Pseudomonas. There could be Listeria, which is also one of the pathogen in this group is there, very common pathogen. Fungi, which could be psychotrophs and can cause food spoilage. There could be groups of Fusarium, there could be of Mucus, Penicillium, and Candida, Rhizopus. Now, it is important to see that how we are reducing the temperature and in a controlled manner so that we can address energy efficiency on one side, and another side, the challenge of food spoilage is also reduced by retarding microbial growth, biochemical changes, and chemical changes. For food, if we see that minimum and optimum temperature, growth temperature for certain pathogens, we can observe that minimum temperature and the optimum temperature range, some of the pathogens, they are able to grow 7 or below, particularly like Staphylococcus aureus in this table. Or if we look for E. coli strains, looking further if for Clostridium botulinum, looking for if it is a Listeria monocytogenes, temperature which is 0 or it could be sub-zero. So there are challenges. It means contrary to the thermal processing, the products which are preserved using low temperature are not really sterilized. So it is very, very important to know it, and in order to deliver a safe food. The principal discussions, if we take in relation to the low temperature, I mentioned that contrast to the heat temperature, low temperature practically not destroying microorganisms or enzymes per se. Both are important agent for spoilage of food. Here, refrigeration is only retarding spoilage, because it is retarding or it is making the cause of change, cause of spoilage just static. It is not microbicidal, it is influencing the growth of microbes, action of enzymes in a way that is arrested to some extent. It may still work, means it may still be alive, an enzyme may still function during storage, low temperature storage. Depends that what temperature is there, what product is there. So, starting material should be of a high quality, and a definite shelf life, extended shelf life will also depend on the initial quality, because initial quality cannot be amended if a pathogen contamination is already there, and if it is not taken care in terms of the pre-treatments, it will remain with the food. So, and even post-processing, every care has been taken in terms of the quality, in terms of the processing. Important to maintain a reliable cold chain. Reliable cold chain means no temperature drop. The range of the temperature should be strictly maintained. And in a way, we can say that it is a hurdle approach. Means we are combining two concepts, like pasteurized milk. It is pasteurized, which is thermal processing. And then it is put to the temperature 4 degrees centigrade or below. So this is a combination of low temperature with other methods. Reduction in terms of the temperature, if we see that how it is affecting enzyme activity also. Here the figure what indicates, if we take our product, we keep our product in the refrigeration zone or in a lower temperature. What temperature it should be, we'll discuss it little later also. So considering it a normal refrigerated temperature, which is a domestic refrigerator can also give, we are able to see that enzyme activity and enzyme stability, both are depicted here. The enzyme activity, which gives a plateau kind of change or behavior here. So initially, the activity of enzymes till it is refrigerated, you are able to see it remains controlled. Slightly activity may go over time. But if it comes out of the temperature, temperature increases, there is a sharp rise of activity. And then when temperature go beyond, theoretically the activity should have go on increasing, or it should have taken a exponential kind of behavior. But enzyme is protein which is having its relation to a higher temperature, so protein denaturation leads to. So that's how again the activity comes down. So this is how the enzyme activity is also related to the temperature, and particularly with low temperature. Low temperature preservation is mainly employed or commercially employed in form of chilling or freezing. So they are two distinct processes. Chilling is referred to, usually to a temperature if we employ 0 to 8 degrees centigrade, that even 8 degrees centigrade or below we can achieve in the domestic refrigerators. And up 8 going low means coming lower from 8 degrees centigrade, then we can go up to almost freezing point of food, some of the foods or water. Here, important to see, because we have seen that mesophiles stopped around 7, but the challenge of psychrophiles is still there. And in this psychrotrophs, we have seen there were certain pathogens. And if there is a product which is like a meat product or so, then the challenge, we need to see that the temperature which might require in chilling could be little sub-zero. So that is how the chilling is defined. While freezing is, of course, the temperature is needed which is much lower than 0, and conventionally a temperature which is used as a storage temperature minus 18 degrees centigrade, though freezing might take place even much lower than the 18 degrees centigrade, minus 18 degrees centigrade. And low temperature applications is one thing as storage and processing, if you take it, then the frozen foods. Even some other applications are also associated. Just I'll quickly cite them. Say, for example, hardening of the fats, butter, freeze concentration, freeze drying, air dehumidification, ripening of cheese, that is basically aging of cheese or aging of wine, meat, improvement in the meat and bread cutting. Breads are cooled before cutting, so that the cutter, means the bread or the starch, should not adhere to the blade. In soft drink industry, the mixing of CO2 done at low temperature, that increase because of increased solubility of the CO2. That's how low temperature has different applications associated with the food. Particularly if we look for chilling and freezing, and first starting with chilling, the first application which comes in mind is the cold storage. That is applied to the fresh harvest. So chilling fresh foods or storing fresh foods at low temperature, what we need to remove the sensible heat. That is also called field heat. And there is respiration which is continued. The harvest is still having the cell activity, respiration is going on, and that respiration needs to be taken care for cooling, the heat it will generate. Say, for example, if just going back, rate of biochemical changes, whether it is microbiological or enzymatic, or they means the changes will increase logarithmically with temperature if the temperature is increased. So the reverse is also true. If the temperature is reduced, so that comes down. So chilling is used to retard respiration. We have mentioned in our discussion also. And going further, the respiratory heat, if we see at 20 degrees centigrade and at atmospheric pressure, that comes in order to the 2.8 into 10 to the power 6 joule per kilo, joule Kelvin per mole. Why? There is molecule of glucose that is being oxidized, and that is how this exothermic reaction is taking place. So means in a cold storage structure, what do we need to take care for total heat load of the cold storage? Heat of respiration, sensible heat of the containers, of the food itself, heat which is being evolved by operators if there is a movement within that zone, heat loss through the roof and walls, though there are insulations provided, but definitely it is not totally inert, and heat loss which could be through the floor. So that is how all accounted, and that is how the cold storage is designed. For low temperature or the cold storage of the fresh harvest, it is very important to see that how do they behave, or the respiration or the respiratory rhythm which is of the harvest. If there is particularly an optimum temperature of storage is identified for each commodity, each fresh produce. Temperature which goes above it, then there could be deterioration because of over the time. If we see that at a particular temperature, so means over the time by and large it remains same. If we see up to a limited time unless the commodity is entering into the spoilage, but the temperature, if it goes up, the respiratory rhythm is also going up. So respiratory rate is increasing. Here it is important to note that one thing, there is an oxidative change, respiration is taking place, but also important that with temperature, transpiration may also take place, means loss of water from food, because water is being produced and that water may come out. And so eventually we'll lose to the, like shriveling of fruits or apple you would have observed in refrigerators. So that is leading to a senescence and aging of the food commodity, which is stored at low temperature. Key factor for shelf life of fresh crops in chilling storage: type of food, variety of cultivar, which part of the crop we are selecting, sometimes a leaf, root, or a fruit, which could be actual biological fruit. So means their requirement of chilling would be different. Condition of food at which it is harvested, is there any mechanical damage, what was the maturity at which it is harvested. Like leafy vegetable, we harvest at a stage which is quite tender stage. For fruits, just matured stage we harvest. Then the temperature of harvest, storage, means before you are lowering down a temperature, how long it has been exposed to a field temperature or a elevated temperature. Storage conditions, what are the conditions maintained in the retail supply chain. RH is very, very important, because the transpiration loss, that is also connected with RH. If RH is kept high, the transpiration loss would be lesser. Now here for fresh harvest, we basically classify the group of fruits and vegetable, the one which are stored between 0 to 4, another 4 to 8, and then there are few which are requiring above 8, I'll say even above 10. Here a glance can be taken that commodity to commodity, the temperature requirement, relative humidity requirement may vary. And even there is a range. Say for example, if you take, there is a range for lime, there is a range for, particularly if we see banana. So means within that range also storage life will vary depending on what temperature you have employed along with the RH. That's how there is a range of storage is reported in this particular table. So means it is important to know and work out the conditions with certain initial trials depending on the food commodity, fresh harvest, along with its cultivar and variety. So it will go up to that. Requirement of effective cold storage or low temperature storage will be mainly a controlled low temperature, because it is low temperature storage, along with the air circulation, because food is generating heat. So are we able to take that heat through air circulation quickly away from the food or fruit or fresh harvest? Humidity, that is RH. Then the gas atmosphere is also sometimes it is played with to control the respiratory rate. In terms of behavior of fresh harvest, particularly fruits, there is two terms. There are two terms which are very relevant to highlight here. The behavior one is climacteric, another is non-climacteric. The fruits in particular given a classification of climacteric and non-climacteric both. However, vegetables, they normally respire similar to the non-climacteric fruits. So, say for example, climacteric behavior, the respiration which is on vertical axis along with the time. So, there is a definite behavior. If you compare with non-climacteric, you see the respiration or the respiration rates, there is downfall. However, in climacteric fruits, in the storage, keeping conditions constant, there is a sudden rise. So, there is a climacteric peak at which they come. So, for that region, the spoilage is faster even at a particular storage condition. And they have a limited shelf life. So, that needs to be addressed and that needs to be taken care. But there is advantage in terms of climacteric fruits. They can ripe fully if they are harvested at the completion of their maturity. So, means we can have a period, they are harvested under ripening or means unripened stage, but matured stage, and then the transportation can be carried. While for non-climacteric fruit, the scenario is different. They cannot ripen off the plant. The cold storage, which might induce certain undesirable change, also one has to be aware about. So for the learners here, it is important to note the low temperature may be associated with the changes like cold injury, which may eventually lead to the loss in ripening. There could be loss of certain nutrients like vitamin C. There could be changes which could be like carbohydrate compositions. Learners must have observed sweetening of the potatoes immediately after cold storage. If they are taken off, the potato lot is taken off from cold storage, they gives some sweeter in taste. And here it is important to see that chilling threshold or the lowest safe temperature has to be worked out for each of the commodities. If we want to minimize these undesirable changes, if we want to address these challenges. Chilling injury is important, so I am here highlighting it in detail. Chilling injury is associated with a damage to the plant part, which is caused by a low temperature, which is in the chilling zone. So, means in the refrigerator or cold storage kind of zone. So, that's how it is above 0 degree centigrade. And this is more prominent for, we have more tropical, subtropical fruits with us. So, for those kind of commodity, it is more important that those commodities are more susceptible. Why this takes place? Because tissue got weakened, and then because of it, there is some undesirable changes are taking place. They are unable to carry or the normal metabolic processes. That is the basic reason for, because we are maneuvering metabolic process. And that will lead to the physiological and biochemical alterations. And that is how you can see that an apple which is shown here is giving an indication of chilling injury. General symptoms which could be seen in the chilling injury, they are on the surface, like here in case of apple we are seeing. There could be surface pittings, mostly like commodity tomato. Development of off flavors could be there. Failure of ripening could be there in some of the fruits if there is disturbance, very high disturbance in the temperatures. Temperature abuse is of high grade. Incidences of the surface mold growth may also be seen little later after a decay. Here the susceptibility of different fruit, which is shown. Learner can take a pause on this particular table and can look into the kind of change, the generalized change we have discussed. They can note it. They can note that what is specific means, though we go for a low storage temperature which is below 8, but banana we cannot store below 8. Lemons, that is not the preferred temperature. Mangoes, they also need higher temperature. So there is every commodity, every single commodity of fresh produce category needs a particular temperature range to minimize these changes. And here temperature is not alone the culprit. Varietal changes, time at subcritical temperature exposure means disturbance in the cold chain, metabolic state of the tissue, atmosphere, which is including RH and gas environment. And it is not necessary, only certain fruits will show this behavior of chilling injury because they are chill sensitive. Another thing which is vitamin C loss, it is depicted here that the same commodity, if we look in terms of the temperature and the number of days, the loss percentage will vary. So there are table which is showing that there is loss of vitamin C. Another study which is reported in the red text, it indicates that almost no loss in vitamin C even during a 6 month of storage, and the commodity is Washington apple. So means the loss will also depend on the commodity. And now we'll discuss low temperature storage of meat and meat products. Though the meat is not a living organism, but important here is to control the impact and effect of enzymatic activity, proteolytic activity. This particular process is retarded using low temperature. Different parts of meat, they may offer different resistance to cooling because lipid content, lipid distribution may vary. Rapid cooling in particular case of the milk is very important. And if we are able to go for a rapid cooling of meat surface, it will nearly stop the microbial growth in terms of weight loss reduction, in terms of minimizing discoloration. For here, the learners must note that relative humidity for meat storage is also kept high. That we have seen already for the case of fresh harvest. In meat, what is most important is safety. It is more susceptible to the microbiological changes and the safety issue. It is must for the meat products that the chilled state should be maintained throughout the supply chain until the meat is processed and consumed. The challenges in storage of the meat may also be seen. These challenges could be color change, cold shortening, tainting. Color change, if you see the figure here, the meat color, there are pigments. The oxymyoglobin may turn into the myoglobin with the oxygen, in presence of oxygen, and then eventually it turns into the metmyoglobin. So it means the meat, which is slightly brownish fresh meat color, turning to brown. Cold shortening, that is a change or the shortening or the textural change in the muscle. And the meat may turn into a rubbery kind of product. And for this, if muscle temperature is falling below 12 degrees centigrade while the muscle pH is still 6, then this challenge may come. To prevent this, what is important is giving an electrical stimulation to reduce the pH rapidly, onset of rigor, that thereby minimize the incidence of cold shortening. Tainting is taking off orders if it is not properly stored or stored with other meats or odorous fruits or vegetable. chilling methods, if we see which are employed for solid food stuff, usually the commercial chilling is based on the air systems, means air circulation, chilled air which is being circulated. There could be different design for it. And circulation is made using fans. Fan draws air through the cooling coils, and excessive dehydration can be a challenge here. However, there is an advantage in terms of a fast cooling, or they could be versatile in their applications. Normally maximum air flow which can be taken 6 meter per second. There could be impingement, can help for minimizing that loss of moisture, and faster cooling can also be carried through this. Air system can be used for both freezing and chilling. For cold storage, what is important to see that compatibility. I mentioned just now that tainting in meat. Similarly, fruits and vegetable needs to be seen. This is a table which shows compatibility match of the commodities, whether they can be stored in the same temperature or same chamber or not. Like apple and banana, N. So means cross action will take place, so it is recommended not to go for it. If it is apple versus grapes, then it is Y. No cross action, yeah, this combination can work. And these are available through the agencies which are available in the countries. They have published a lot of literature that can be referred, like National Horticulture Board has published manuals. Now, coming to another aspect of low temperature, that is freezing. Freezing is very energy intensive process because the phase change is involved here. And in this thermodynamic phenomena, what is important to note, the high specific heat of water, high latent heat of crystallization. So that's how it is energy intensive. And freezing needs nucleation as a prerequisite. What is nucleation? That is first ice crystal forming as a seed, and over which the crystal grows in size. And thereby this nucleation will serve as the initial process of freezing and critical step which will make the freezing complete. A pure water may go unfreezed even up to minus 42 degrees centigrade. So what is important for nucleation? And energy and extra nucleation effort that is required for freezing process. In foods, normal water, say for example, which we know that freezing point is 0 degree centigrade. But for the foods, if water is there, that depends on that what solutes are there, what form of water is there. Water could be bound water or a free water. Bound water will not enter into freezing. Free water is more available for freezing. So lot of water may remain unfreezed even at around 0 degree centigrade. So means we need a high rate of heat transfer. The difference of the temperature, the minus temperature side and the product side, that difference should be larger. So the temperature at which the first ice crystal will appear, that is commonly regarded as the initial freezing point. We can see the freezing, how it takes place in the food. So for example, initially, some of the ice crystals are forming, if you see this figure. Then gradually the number of ice crystals are increasing. And if we look this in terms of the time and the temperature of the food, you see that there is initial cooling. Then when it is coming sub-zero, there is a state of supercooling. That is marked as AS. Then slight increase in the temperature. Then thereafter the freezing starting, mainly freezing, particularly if you see that B to C, that is a rate of heat transfer is almost constant. And major part of the ice is being formed. Further, that you can say that a dip is observed, and then after E, if you look for EF, that's the temperature of ice water mixture is falling, and the temperature of the freezer is that product will try to come closer to that. So that is the cooling. Here, if you see the temperature versus the percent of ice form, that is shown in this. Gradually, the temperature is decreased and the water, which is unfrozen water, is also decreasing. What is important to look into this freezing process to have rapid frozen foods, because slowly if we'll freeze, then the ice crystal would be of larger size. That would be damaging to the product. That will also be important in terms of the texture loss to the product. If the overloading of the freezing is there, then the crystal size may also be affected. While freezing the food, it is important to note that what is actual rate of freezing, which has to be rapid, and where the ice crystal is actually forming, whether it is extracellular or within the cells, because this will cause impact on the food quality. Quick freezing will lead more or less uniform freezing in extracellular and intracellular water. Otherwise, the concentration gradient will come that will impact the quality of the food. In freezing, what is important to note that concentration may adversely affect the quality of the food. And damage may be there due to the larger ice crystals. Further, if we see that certain chemical changes may also eventually take place due to the concentration effect. So, in terms of low temperature, what we have today discussed, initially we discussed the low temperature storage, which was ever freezing temperature. That was mainly employed to the fresh harvest and also to the meat products. The same fresh harvest or the meat products can also enter into the freezing, where the temperature goes below zero. But here, it would be important to note that high energy intensive process, but eventually will give an extended shelf life. So, low temperature is very, very important in the current scenario. We see that almost every house, you see that there is a domestic refrigerator available. There are ready foods available which are offered through the freezing. That's how we extend the shelf life using low temperature. Thank you.